Antibody-drug conjugates, or ADCs, are a class of anti-cancer treatments that are designed to leverage the specificity of antibodies to selectively deliver potent cytotoxic therapies to tumor cells while sparing healthy cells. XB002 is an investigational ADC comprising a high-affinity human IgG1 monoclonal antibody targeting tissue factor conjugated to a potent, cleavable linker cytotoxic payload, Zymelink auristatin, or ZLA. Tissue factor initiates the extrinsic blood coagulation cascade in response to external trauma that causes a breach in the endothelium. Tissue factor is primarily sequestered within intracellular pools in subendothelial cells with only limited membrane expression. Vascular injury exposes tissue factor to coagulation factors. Tissue factor is mobilized from intracellular pools and trafficked to the cell membrane, initiating blood coagulation at the site of vessel injury and activating factor 10. Activated factor 10 is released and triggers the completion of the coagulation cascade, resulting in the formation of a fibrin clot, thus providing a hemostatic barrier to limit hemorrhage. Many solid tumors aberrantly express high levels of tissue factor on their cell surfaces, which may be associated with poor prognosis and disease progression. Metastatic cells at the invasive edge of tumors prominently express tissue factor at levels as much as 1,000-fold higher than non-metastatic cells. Through aberrant non-hemostatic signaling, tissue factor may promote tumor cell proliferation, cell survival, angiogenesis, and metastasis. Tissue factor is a candidate target for ADC therapy because it is preferentially expressed on tumor cells compared to healthy cells and undergoes rapid internalization, ensuring efficient delivery of the ADC into the tumor. A challenge for tissue factor-targeted ADCs is that they can potentially disrupt the essential hemostatic function of tissue factor by interfering with its binding to factor 7, which could limit their ability to provide a clinical benefit. However, XB002 is designed to bind to tissue factor with high affinity without interfering with extrinsic pathway-mediated activation of factor 10 and blood coagulation in preclinical studies. Binding to tissue factor on the tumor cell surface may trigger rapid internalization of XB002, which is then trafficked to the lysosome, where the ZLA cytotoxic payload is proteolytically cleaved. The released ZLA cytotoxic payload has been shown to block microtubule polymerization, inhibiting cell division and inducing apoptosis of the targeted tumor cell. In experimental human xenograft tumor models, XB002 treatment is believed to inhibit tumor growth and may lead to tumor regression. The anti-tumor effects of XB002 may also be due in part to IgG1-mediated effector functions, including antibody-dependent cellular cytotoxicity and antibody-dependent cellular phagocytosis, as well as immunogenic cell death. Tissue factor has emerged as a candidate target for ADC therapy. Exelixis is investigating XB002 as a single agent and in combination in advanced solid tumors.